Hello and welcome to another lecture of the course Aquaculture. Today we are going to start a very interesting and very important topic, um, induced breeding. Why this is so important? Because without having good quality seed, without having good quality fish seed in desired numbers of desired quality at the desired time, we cannot start aquaculture practice. So, briefly, we will discuss what it is, how it is practiced. Induced breeding is a technique whereby ripe fish breeders are stimulated using pituitary gland extract or any other inducing agent to breed. In fact, whatever breeding conditions are required by the fish in its natural condition for natural breeding, uh, we try to simulate the conditions, both external as well as physiological by inducing some kind of hormones or injections, so that fish can breed in confinement. This is especially very important for the fishes which do not breed in confinement. For example, Indian major carps, the katla, rohu, and mirgal, they do not breed in confinement. They need flowing waters, riverine conditions, flooding conditions for breeding. So, in ponds, we do not have those conditions. So, they may mature, they may show ripeness, but they will not spawn. So, to ascertain that the maturation is proper and spawning takes place, we will have to simulate the conditions and induce the fish so that it can breed in confinement. Hypophysation is often referred as a process in which ripe fish brooders are induced to breed in captivity using pituitary gland extract. Now, a brief history of how all this thing started, especially in country Argentina. It was Housey in 1930 who prepared pituitary extract for the first time, and he used that in viviparous fish to cause premature birth. But that gave the concept. And then Brazilians in 1934, what they did, they were successful in carrying out induced breeding in fish using pituitary extract. In India, in fact, it was Hamid Khan in the year 1937. He used Syranis mrigala to induce breed. He tried to induce breed it by using mammalian pituitary hormone, but he was not completely successful. The eggs were produced. Success was to that extent, but they were not fertilized. Again, one ice breaker was there that you just tried it. Then came Dr. Hiralal Chaudhary, the person who is considered a legend in induced breeding in India. What he did, he was first to successfully induce breed Syranis mrigala, Syranis reba, and Labiorota. That was a milestone in the development of aquaculture practice in India. And later on, after 1957, then there were many other fishes which were induced bred, and for the induced breeding of many candidate species, technologies, standard induced breeding technologies were provided. What are different steps to carry out induced breeding, especially with pituitary gland? So, what we do, we collect pituitary gland, then preservation and storage of glands, then preparation of extract and its preservation, then selection of brooders, preparation of injection and dosage, spawning. First one is collection of pituitary gland. How should we collect, and from which fish we should collect? It is always advisable to collect the pituitary gland from not very immature fish, from not the spent fish. Try to go for the fish collect it from the fish which has started showing maturity in the beginning. So you can collect pituitary from adult fishes. That will work. For better work, we will have to kill the mature fish. That is again not advisable, not very good thing. And we should avoid the young fishes for collection of pituitary. That will not provide you better result. And it's always advisable to go for fishes which are very close to that genus. And if it is not possible for you, even fishes from distantly related, which are distantly related, that will also be fine, because people have used pituitary from other classes also within the phylum, within the phylum Chordata. They have used pituitary gland of different other classes to breed fish, but it is always advisable to go for closely related fish to get pituitary from. Now, how to extract pituitary out from the fish body? There are two methods which are often used, through foramen magnum and through cutting the roof of the skull. Through foramen magnum, expose the foramen magnum and remove muscles and parts of the vertebrae left of the cut head. Usually, the head portion is cut, and then we try to take out pituitary through the foramen magnum. Remove the adhering tissues or fatty substances, whatever are there. Then detach the anterior part of the brain and remove it carefully. While detaching the brain part, you should be very, very careful. Detach it very slowly, gently. If you are doing it, not carefully, then pituitary gland may go along with the brain, and you will not be able to identify that where pituitary has gone. Many parts of the brain will look like pituitary gland, or pituitary may look like this part of the brain. Now the pituitary is exposed. Clear the membrane and collect it in a petri dish. Here it has been shown. Now, cutting the roof of the skull. This is very common, very common. And what you have to do, you have to simply cut the skull portion, roof of the skull. Then brain is exposed. Remove the fatty substances or whatever other tissues are there. Cut the olfactory and optic nerves and lift the brain carefully. You have to lift the brain, so whatever attachments are there around, you will have to cut and then lift it very, very carefully. Pituitary gland is situated behind the optic chiasma and exposed and collected. Usually in the cavity where it is lodged, if you can use any type of instrument to take it out. I have seen people taking it out with something which is like a very small scoop. So if you put that in the cavity, then in that scoop pituitary will come out, and then you can collect it in a petri dish or whatever vial you are having. Now, once pituitary is collected, you can collect pituitary the day when you are planning to carry out induced breeding of fish, or you can collect the pituitary and preserve it, so that at the time when you have to carry out induced breeding experiment, you don't have reason to worry about the availability of the pituitary. So you can use fresh pituitary, you can use stored pituitary if it is preserved properly. So how it should be preserved? Freshly collected pituitary gland is used or can be stored for future use, as I have just told you. The properly preserved pituitary gland retains its potency for several months. It is always advisable not to go for very old pituitary. Try to use it in as fresh form as possible. Even if you have to use it in a preserved form, try to get information on when the pituitary was collected, and what kind of fish was the donor fish from which pituitary was collected, and how it was kept while preservation. I will tell you that the conditions, freezing conditions, information regarding freezing conditions are very important. Following methods can be used to preserve: freezing, then you can use absolute alcohol, you can use acetone also. The pituitary gland can be immediately frozen. As soon as it is taken out, we can immediately keep it in a frozen form. Or the other method is in absolute alcohol. This is very common. The collected pituitary gland is kept in absolute alcohol in airtight vials for 24 hours. For 24 hours, first keep it for 24 hours, then the alcohol is changed and vials are kept at room temperature or in the refrigerator. You will have to keep on periodically changing the alcohol and keep it at room temperature. You can very well keep it at room temperature. No refrigeration is required, no freezing is required. The alcohol should be changed occasionally from time to time. Maintain the gland in good condition or for a long time. That you will have to take care that the conditions are met for proper preservation of the pituitary. That will ensure the potency of the pituitary. Otherwise, your breeding operation may not be that successful. For preparing the extract, pituitary gland should be dried for 2 minutes at room temperature and then weighed, because you are taking it from preservative. So let that evaporate, and maybe you can keep that on some blotting paper or something, filter paper, and when it is dry, then weigh it whatever your requirement is, and then go for preparing the extract. And here you will see different sizes of pituitary glands of Katla. Katla has been shown. You know, we should have information on for what time they have been preserved, and what was the procedure for their preservation, and what could have been the potency now. So only after that you should go for this. I have personally experienced that pituitary which has not been kept properly and which are very old, you do not get good results. Especially if you are buying it commercially, then there are great chances that whatever pituitary gland you are getting, the success rate may vary. Maybe if 100 pituitary gland you are getting from somewhere, maybe 10, 15, 20 will work, others won't. Then how you are going to know that? So that is problematic. That is why I say that, okay, if you have to use pituitary gland, try to get it on your own farm, and then you can use it. You can preserve it in acetone, or you can preserve it on your own, because that is not very difficult. So if you are preserving it on your own, you are changing the alcohol, you are keeping it at moderate temperature, then you can use it. Collected gland is immediately kept in fresh acetone or ice chilled acetone in vials for 36 hours. You can use refrigerator in case of acetone, but even if you are not using refrigerator, and if the room temperature is not too much, 20-25 degree, then it will be fine. During this time, the acetone should be changed at an interval of every 12 hours. But while using acetone, try to keep the acetone preserved pituitary gland at lower temperatures in the room. If it is not possible, go for refrigerating it at 10 degrees centigrade. For preparing the extract, the gland should be dried for 1 hour at room temperature and then weighed. Now we have got pituitary, whether fresh or preserved. We have to prepare extract, and extract may also be preserved. Previously weighed and preserved glands are taken out and homogenized in distilled water or 0.3% saline. It is preferable that if you use saline, it is considered better. Homogenized gland then diluted to the desired value. First you homogenize it properly using 0.3% saline. The dilution is made at the rate of 0.2 ml per kg body weight. This is critical. People don't take care that how much saline we have to use for preparing the extract. So if you pour too much of saline, then you cannot inject all the suspension pituitary gland extract to the fish. So that depends on the weight of the fish, that how much. Here it is written 0.2 ml per kg body weight. This should be the ratio that while preparing the pituitary gland extract. The suspension is then centrifuged after homogenization. It is centrifuged, and we know that in centrifugation what happens, it is rotated at a very definite speed, and when you will do that, then debris will come down and supernatant will be on the upper side. The supernatant is then used for injection. For preparing the pituitary extract beforehand in bulk, the extract is prepared in distilled water and preserved in glycerine. Glycerine we can use to preserve at the ratio of 1:2, distilled water 1 and glycerine 2. Then you can keep your extract there also. A concentration of 20 to 40 milligram of gland in 1 ml of water and glycerine is considered most convenient. So these are just recommendations. If one person has got this concentration very convenient, you may at your place, because the conditions at every place varies. The condition of the brooder, the condition of the climatic conditions, the equipments you are using, the reagents you are using, the chemicals you are using for doing all kind of work, the variety of fish you are using. So everything, all the factors may be different from one place to the other, and human error, if somebody else has done, there can be different kind of error, and when I am doing, there may be different magnitude of error. So it is always advisable to standardize the procedures for your farm, for your region. The pituitary extract can also be preserved in propane and stored in refrigerator for up to 30 days only. So it must be taken into account. Or we can also use TCA, 1.5% TCA, trichloroacetic acid, and store it in refrigerator. So that was regarding preparation of the extract. Then we have pituitary gland suspension of katla after homogenization. You can prepare pituitary gland suspension by homogenization, and then this homogenate will be centrifuged in order to get debris and supernatant separated. Then comes selection of breeders or brooders. Healthy and ripe brooders should be selected, and in case of Indian major carps, we should select it between 2 to 4 years of age group in order to get good fecundity, good fertilization success, and all other parameters which are related to fish breeding performance. You will get all these parameters in showing very good performance. Ripe female, identified by smoothness of the dorsal surface of the pectoral fin. Abdomen is soft, bulging, rounded abdomen with swollen and reddish vent. So that is how we identify. In fact, in case of Indian major carps, you don't have to do much thing. You simply take the brooder. The belly of the female will be swollen, and you can just by looking at it, you can tell that whether the fish is ready for spawning or not. And if you take the belly on your hand, on your palms, by its belly of the fish. If you take the fish by its belly, then when the pressure will be applied by your palm on belly of the fish, some of the eggs may come out. If that is the stage, the running stage, that is one of the very important stages for selecting the spawners. If that is the case, go for it. You will be getting success in that. Then ripe male. You can identify that during the breeding season you don't have that round bulging belly, and then the dorsal surface of the pectoral fin just near the opercular opening, gills, near the opercular opening, the pair of pectoral fins. Inside of that pectoral fin will show roughness. If we will just try to feel it from the inside of towards the body of the fish, that will show roughness. But in case of females, it will be very smooth. So that way also you can identify the male and female. So and here again, if you will take the fish by its belly on your palm, then here the gametes in the form of milt may start coming out. So if these stages are often referred to as running stages. So if you are having fish brooders in running stages, then that is the excellent stage to go for induced breeding. You will get success. Female laborata and male laborata, it is shown that how they look like, how their vent look like, how their belly looks like. Then here also they have tried to show, Katla brooder, again the carp. In case of, catfishes, the condition is little different. In case of female, you will get bulging belly, but here what we do, we identify the male and female not every time, because they are not, these catfishes are not very high fecund fishes. They do not give large number of eggs. They may be producing relatively less number of eggs compared to carps. So their belly may not be as bulging or swollen as in case of carps. So what we do, we identify the fishes by the papilla present on the vent region. In case of females, the papilla will be round button shaped and maybe slightly pinkish in color, pinkish to reddish in color. But in case of males, it will be pointed. The papilla will be pointed and very light color compared to female vent. Preparation of injection and dosage. Now, when we are using pituitary gland, then how dosage are to be calculated, how injection is to be prepared so that we can give it to the fish. To determine the quantity of gland to be injected, the selected breeder should be weighed, and one set of brooders in case of Indian major carps, when we are going for this traditional breeding program using pituitary gland, then one female plus two male comprise one pair. Injection is preferred to be given on a cool, cloudy day in the evening when the temperature is low. What I told you in the beginning, that we try to simulate the conditions what are available in the natural environment, rainy season, flooding, and so those conditions, if we provide them, then they may show good response. So injection we can give intramuscular or intraperitoneal. Intramuscular, caudal peduncle or near shoulder region, you just simply lift the scales and you can give injection to the fish. Then intraperitoneal at the base of pelvic fin or pectoral fin. If you just lift the pectoral fin or pelvic fin, just at the base, inner base of the pectoral fin, you will find that the portion is very delicate. At that portion, just insert the syringe very gently and give the injection whatever is required. So these two are very common methods to give injection. What is the dosage? Preliminary dosage, in case of females, we while using pituitary gland, we give two doses. Preliminary dose 2 to 3 mg per kg body weight. When we start in the spawning exercise, induced breeding exercise, then 2 to 3 milligram of pituitary gland per kg body weight of female brooder is given, usually at the sunset time, just before the sunset time, and then second dose is 5 to 8 mg per kg body weight, 5 to 8 mg of pituitary gland per kg of female body weight after 5 to 6 hours of first dose. So if we have given injection at 5 o'clock in the evening, the first dose, then second dose will be at 10 or 11 in the night. Then injection for male, here preliminary dose is not given, only one injection is given, and that is only 2 to 3 mg per kg body weight, and that is at the time of giving second injection to the female. Now, after injection, a set of female and two males, the one set, are released into the breeding hapa or cemented cistern with water circulation. Whatever kind of system, breeding tank you are using, whether traditional hapa system or concrete tanks, there you can release these fishes. Hapa is, in fact, I showed you in my earlier lecture that how bamboo poles are there fixed in the pond, and through those bamboo poles we can fix the hapa we can fix a muslin cloth enclosure, uh, which if we require we can cover that also using some kind of tying that lid or whatever opening you wish to, and this is kept just above you know, the ground, pond bottom, and the space between the hapa bottom and the pond bottom should be at least 1 and 1.5 feet or so, or more than that, depending on the depth of the pond. Here you can release the injected fish, or you can go for tanks also. You can release them in tanks. Then if you are using hapa, then you should take care that the cloth you are using to make the hapa, that should not have pores from where the eggs can go into the pond outside the hapa. Usual size of hapa 3.5 meter, 1.5 meter, 1 meter. After 2 to 3 hours of injection, the breeders become excited and restless and swim actively. Within 3 to 6 hours of the second injection, spawning occurs. So 3 to 6 hours, that means early in the morning, you will get to see that these fishes have bred and you can get the fertilized eggs. Examination of the eggs. To understand whether fertilization has occurred properly or not, you can observe the eggs, crystalline, transparent, like a pearl, very shiny, very, you know, they look like very happy eggs. They are fertilized. And those which are looking very dull, opaque, whitish, they are unfertilized. So you will have to, at this point of time, you will have to remove fertilized eggs from unfertilized eggs and go for only fertilized eggs. Otherwise, the unfertilized eggs may cause different kind of problems like fungal infection, and that can destroy our entire fertilized eggs also. So you will be at loss. Here, from some references I have given you here, some of the eggs I have tried to show. Then dry method or stripping method, that was for the fishes to breed naturally within the hapa. But dry method you can also use. The fishes are examined 3 to 4 hours after the final injection. What happens when we think that, okay, we are not getting very good fertilization percentage in the natural breeding? Why we are not getting very good fertilization percentage? Because the fishes are showing external fertilization, and when the gametes are released, then there is dilution within the water, and there is very short duration of time for which sperms are active. If during that active period time, if sperms do not fertilize the eggs, then fertilization success will not be very good. So in natural environment where there is dilution, where this is taking place, fertilization is taking place, the fertilization success is quite low. So to overcome that, what we do, we keep on observing that after second injection, after what time the fishes may be showing the sign of releasing the eggs. Just at the time when they are showing the signs that they are going to release the eggs, take them out. Take them out, and what you can do, you can have some kind of tray, dried tray, clean dried tray, and what you can do, you can have some feathers also, bird feathers, again clean and dried. Then take some towel, and one person can hold the female in such a way that its vent, the belly region, is towards the tray, and one person can hold the male, the head portion or mouth portion can be held within this arm, and again the belly portion, vent region, facing towards the tray. Now the eggs, one person will press the belly of the female, so eggs are released. Another will press the belly of the male, so that milk is released. Then someone can mix that up, the gametes, without adding water. It is all occurring in dry condition. And when all the eggs are exhausted, when you have got sufficient milk to mix, maybe one male, or sometimes you may need the other male also. But usually one male can do the milk required by one female fish eggs. So once they are mixed thoroughly, then add some water in the that tray or trough and keep it as such for a while. Then try to mix it with the feather. Fertilization will take place. After fertilization has taken place, then what you can do, you can wash these eggs to get rid of any extra tissue or something, or some unnecessary things. Then transfer it to the hapa for water hardening, and then to the hatching hapa. So that is the process that I have just told you. You can read it also, but I have explained it with my own experience. Here they have shown how not carps but some other fish. They have shown that how the fishes are stripped. Now hatching. Now the clean fertilized eggs start swelling. The process is called water hardening. These can be put back inside the hatching hapas for water hardening for 5 to 6 hours. Water hardening purpose, you can keep them. Then the hatching of the eggs is complete within 14 to 18 hours when the water temperature is between 27 to 30. The hatching time depends a lot on water temperature. So water temperature may have very strong influence on what time the hatchlings take to come out from the egg. For example, there is one time interval between injection and stripping of the female catfish, and the time interval between fertilization and hatching of the eggs in relation to temperature. So, time between fertilization and hatching, if you go through here, as the temperature increases, the time decreases. So, that is something at higher temperatures, the fish may show, may require less time for hatching. Hatchlings pass through the inner hapa. I will show you in the next slides, I think, to the outer hapa, and the inner hapa is removed. Now, if you are using hapa system, if you are using circular hatchery system, then again the same concept is utilized, but in tanks. So there also you can get the hatchlings. Now the hatchlings remain inside the outer hapa, and 2 more days till their mouth is formed, when these are called spawn and ready for stocking in prepared nursery ponds. The hatched larvae feed on the yolk sac as their food resource. All the hatchlings, the hatched larvae, they have one sac attached to their ventral side of their body near the head region, that is called yolk sac, and they feed upon them until they are ready to get food from outside. After 3 to 4 days, the yolk sac will be absorbed and hatchling will develop, which requires zooplankton or live food for their development. This is how hapa looks like. This is outer hapa, this is inner hapa, and in the inner hapa you have to put the fertilized eggs. From these fertilized eggs, the hatchlings will come out, and ultimately these hatchlings, through the pores of the inner hapa, here it is not shown properly, but the material used in inner hapa is slightly having very similar to mosquito net, and through its pores, the larvae or hatchlings, they go outside the inner hapa and they remain in the space between inner and outer hapa. But at some places only netting material is used, and the hatchlings may go directly outside into the pond, but that is not very advisable, because they are not ready to compete with outside environment at that point of time. So this one is good. You can remove the inner hapa after hatching has taken place. Hatchlings have gone into the outer hapa, and the debris is there in the inner hapa, and you can throw it out, and then you can collect the hatchlings from the space between this. There are different type of hatchery systems. And nowadays, an integrated system is used, circular hatchery hapa, or there are carp hatchery unit consists of a source of water where in situ filter can be kept, a pump, storage tank, aeration facilities with an air blower, compressor, vertical hatchery jars made of glass, there are different type of that will require a different lecture, that how um, the hatching technique has evolved, how different type of hatcheries have come out over a period of time. So there are number of hatcheries that you can discuss, and um, some of them may be discussed here, like traditional one you have studied, then CIP-DAT1 model, and many other models, jar hatchery, and many other hatcheries are there. But nowadays, the most important, the most common hatchery that is being used is circular hatchery. Here, what we do, we have separate sections of breeding and hatching eggs, and large volume of water is necessary because you have to provide the conditions required for their breeding. It is an open system wherein temperature is not controlled, combination of groundwater or canal water. Very similar to Hapa, the concept of Hapa is utilized, but in a different manner. In outside the pond, we have to do everything, you can say indoors. Gravitational force is normally used for the eggs to get into hatching cistern. In fact, we prepare circular hatcheries in such a way that the breeding tank is at an elevated, and so that whatever fertilized eggs are to be transferred to the hatching tank, they will come automatically through gravity in one collection tank, and from that collection tank they may be transferred to the hatching tank, maybe manually, or the one pipe may be connected to that hatching tank also, so that will automatically go to the hatching tank. And from hatching tank we can have a connection of pipes for letting the hatchlings go into the nursery. So as per your own requirement, you can work on. Here the hatching Hapa has been, circular Chinese hatchery has been shown. Here the inner jacket is there, and the outer portion, and the inner portion is having a framework of maybe iron or steel, and over that steel framework or GI framework, you can have one net covering that framework. And so, and in this portion where the water is there, the water is poured into the tank in such a way that it is always in the circulation at a required speed that you can decide, and the so that the eggs, fertilized eggs, do not settle down, because if they will settle down they will perish. And by having by being in the motion and getting in touch with the moving water, their oxygen supply will also be taken care of, and ultimately they hatch out, and we have they do not go outside the tank through this outlet, because this is outlet. So we can complete the hatching process, breeding and hatching process, by in a circular hatchery. Nowadays that is very efficient method, and we can get very good results from circular hatchery. We can breed a large number of brooders at the same time. So that is, I should say, more productive, economical. If you are doing at a large scale, it is quite economical, and you can produce fish seeds of the desired species in desired numbers, of desired quality at the desired time. So thank you so much. We will discuss some other topic in next lecture. Thank you.